So dear friends, welcome. Next lecture in the series is Microbial Spoilage of Food. Having seen the beneficial aspects of microorganisms in food, we now move on to the harmful effects of microorganisms in food. When microorganisms are present in the food, basically food itself, it serves as a nutrients for the microorganisms as it serves a food and energy building material for our growth. Food, it contains majorly, we have seen, carbohydrates, proteins, vitamins, lipids, and all this in combination are metabolized by cells. The cells inside our body, the stomach cells, the intestinal cells, blood, wherever we metabolize the food into energy compounds, which becomes the building blocks and determinantal for our growth. Similarly, when microorganisms are present in food, they also utilize these carbohydrates, proteins, lipids, and vitamins, and we have seen that by using this they can be beneficial when they are used for food production. But in a already produced food or a already harvested crop or a processed meat or milk product, when microorganisms they lodge on the surface or get inside, then they utilize these components and may be responsible for spoilage or poisoning. So we will see that, but before, we have to understand the various types or groups of food depending upon either the storage or the processing. What is their shelf life? Shelf life means for how much time you can keep these foods without using, before consumption, for how long this can be stored. On that basis you can see the foods, they are of various types. They can be grouped into foods that spoil very quickly or the foods that resist the spoiling. Foods that spoil very quickly, they need to contain moisture. As you can see here, moist foods, the moisture, neutral foods, unrefrigerated foods, and the ground or sliced meat. These are foods that spoil very quickly. We can call it as highly perishable foods. Because the moisture content is high, the temperature is ambient, the microorganisms, they will lodge and they will multiply greatly in that. Foods that resist spoilage, this can be called as semi-perishable or stable food. As the moisture level goes on decreasing, there will be less chance that the microorganisms will grow on that. So dry foods, also fruits, acidic foods, refrigerated foods below 5 degrees centigrade, whole meat, these are semi perishable or you can call them as stable foods. Food spoilage is one issue which is causing great loss to the producer now, or the industry, you can say food industry. Some of the characteristic features of food spoilage are unpleasant smell. As you can see in the pictures, the wine, if it is not of some acceptable flavor, it will be rejected. Unpleasant taste and color changes, texture changes. As you can see the banana over there, it has become moldy, slimy. What are molds, what are slimes, you will see that. Texture change in the texture, the strawberry in the picture you can see, it has become moldy. The color, change of the color, tomato in the slide you can see it has become what you can say, dried off, and some fungus or mold has grown on it. So there are various characteristic features, we can call it as types of food spoilage. As we go on to see the types of food spoilage, that is, a food, it deteriorates, it gets spoiled, and it is a big loss to the economy. There are 3 to 4 types or categories of spoilage. Basically it is all about the chemical changes which takes place inside the food. There is, you have to understand the biochemistry of food spoilage. The basic principle of food spoilage remains in its biochemistry. So the substrate, chemical reactions, chemical compounds which are formed, means what is the substrate for that chemical change. Now chemical change can be induced by light, can be induced by certain chemicals, it can be induced by microorganisms. We will see those things in detail. So the substrate is the food itself on which chemical reactions occur and resultant chemical compounds are formed. Which are the factors which are responsible for this, we are going to see in this section. Now the major causes of food spoilage can be categorized into three major types: physical change, physical changes in the food, or physical food spoilage we can say; chemical uh, reactions which cause the food spoilage; microorganisms which cause food spoilage. In physical, as you can see, temperature, light, mechanical damage, okay, radiations, heat, these are some of the major causes of food spoilage. Chemical reactions, enzymatic reactions are there, non-enzymatic reactions are there. Rancidity, rancid one reaction I will be showing you in the further slide. Chemical interactions of various groups and compounds which results into a newer compound which may be responsible for spoilage. Microorganisms like bacteria, yeast and molds, they grow in the food and they are responsible for n number of physico-chemical changes. I can say physico-chemico-biological changes which I will be explaining, and others which is not covered here because this is a microbiology class, but nevertheless very significant and important. Other insects, rodents, animals and birds, they are also responsible for the food spoilage. One of the most important physical parameter responsible for bringing out physico-chemical change in the food is light. Oxidation of food due to light oxidation. Examples given here are reversion flavor of soybean. If soybean is kept in sunlight or exposed to light, there is a reversion flavor, off flavor is achieved. Sunlight flavor in milk, if you keep the milk in sunlight, or sunlight flavor is achieved, which is not desirable. Rapid loss of riboflavin, vitamin D, E and C takes place in the food if it is kept in light. Greening of potato due to some formation of fats, greening of potato may takes place like this. The potato, if it is exposed to light, then a small amount of chlorophyll is synthesized, carbon dioxide is fixed, acetate is formed, mevalonic acid is converted into cholesterol, and some sort of oily type of greenish covering may be achieved on the potato. This is an example of physical change, physical induced change in the food. Now chemical changes, enzymes that cause food spoilage. There are various enzymes. Where are these enzymes present? These enzymes can be present in the food, fruit or vegetable or the meat itself, or it can be produced by the microorganisms lodging on the food. Now try to understand, if it is by the microorganisms, then that will be a cause studied under microbial changes in the food. But here we are dealing with enzymes which are present within the food as a component of the food, and that can cause food spoilage. Enzymes like ascorbic acid oxidase, which oxidizes the ascorbic acid present in vegetables. Okay, the first one, ascorbic acid oxidase, which is present in the vegetables. Ascorbic acid destroys the vitamin C, and that gives rise to a non-acceptable food. Lipase is an enzyme which acts on lipids. Lipids, these are components of milks, oils, and hydrolytic rancidity is the reaction, spoilage reaction. Means it results into a condition called as hydrolytic rancidity, which is not acceptable. Lipoxygenases are present in vegetables, which is involved in the destruction of vitamin A. Pectic enzymes, they act on pectin present in the fruits, and destruction of pectin results into softening. The tomato picture which you just saw, that can be one of the reasons for softening, that is destruction of pectin, and then due to the pectic enzymes. Then peroxidases results into browning of the fruits. Polyphenol oxidases results into browning, off flavor and vitamin loss in fruits and vegetables. Whereas proteases, these are the enzymes which destroy the proteins present in the foods like eggs, crab, lobster, flour, wheat flour, it contains protein, and that protein is acted upon by proteases. What is the source of protease? That will be a separate aspect to be considered. If it is already present in the food, then that will be the cause. If an organism is present which is synthesizing these enzymes, then that will be a microbial spoilage of food. Here we are discussing the chemical spoilage of food. So reduction of shelf life. Shelf life is how many days, you can see, that is the date of expiry of a food. how many days you can keep it on a shelf before you uh, take it for consumption. Reduction in gluten network is also there due to proteases. Thiaminase, thiaminase is an enzyme which is responsible for oxidation or destruction of thiamine, a very important amino acid which is component of meats and fish. Now enzymatic browning, you can see one chemical change which is taking automatically. If you just cut an apple and keep it in the air for long duration, that is the phenolic substrate contents in the apple that will be acted, oxygen is present, O2 is present by the enzyme polyphenol oxidase, and hydroxylation of monophenol to orthodiphenol and dehydrogenation of orthodiphenol to orthoquinone takes place. This is purely a chemical reaction. No microorganism is involved in it, and therefore it is a chemical induced spoilage of food within by the components present within the food. Here the example, best example of apple, which converts the phenolic substrates into diphenol, orthodiphenol, orthoquinone, which results into browning of the fruit. You can see here in this example an overview of fruit ripening with particular emphasis on textural softening. Proctopectin in unripe mango, kairi. Then intermediately half ripened mango contains soluble pectin. In this intermediate form you can see the biological, biochemical, molecular, organoleptic changes start to take place. From sour to it starts to become sweet, and then it becomes ripe. This is the what you can say normal type of chemical reactions going out in a fruit. So protopectin, soluble pectin and softening, decomposition of protopectin, that is a protopectin is somewhat sour, it gets decomposed when it is ripening, it is automatic taking place within the fruit. And if you allow over ripening of the fruit, then there is senescence and excessive textual softening taking place and spoilage of the fruit takes place. So this is chemical changes. We are not much into studying this, but this is one of the important parameters which is studied in food science and technology related to agricultural sciences, because they have to study harvesting. Here our main aim with respect to this syllabus, I would like to tell there is no end to what you need to study and what interest you can develop. So you should have this interest also. But I will now move on to the microbial spoilage. But before that, there is a thing called as rancidity. Rancidity is production of acids, and it makes the food stuff acidic. So there are two types of rancidity: hydrolytic rancidity and oxidative rancidity. Again I am saying why it is important for you to understand the chemical changes, because these chemical changes can be brought about by microorganisms also. Here in this slide I am trying to explain you the chemical change. Chemical changes brought about by the chemicals present in the food. So hydrolytic rancidity example is triglycerols present in the food. They are acted upon by lipase and it gets converted into glycerol plus free fatty acids. Glycerol will be converted into sugar later on. Okay, but these free fatty acids, which are the resultant compound of lipase activity, gives rise to acidity. A volatile bad odor is always there, but apart from that, the acids are formed and therefore it is called as rancidity. In oxidative rancidity, the example you can say linoleic acid. This is present in nuts, groundnut, etc. That is acted upon by the enzyme lipoxygenases here, and this lipoxygenases is converted into hydroperoxide with the help of the enzyme hydroperoxide lyase into hexanol, and 2-hexanol, 3-hexanol, etc. And this is again responsible for acid production in the foods. So you just remember, this detailing may not be needed for your examination point of view, but for your knowledge point of view, you have to understand that rancidity is a chemical change when it is brought about by the compounds present in the fruit. But if it is by microorganism, it will be microbial food spoilage. Maillard reaction is resulting into brown nitrogenous polymer in the fruit. Food, you can see most of the foods, they get converted into sugary compounds. They get converted into browning. In the last lecture I had talked about caramelization, but that is while preparation of the food we deliberately change the color. But here, due to light oxidation, chemical reaction like D-glucose, the Schiff's base glucosamine is formed and melanoidins are formed. These are brown nitrogenous polymers which give rise to undesirable qualities in the food. Now come to the main discussion point of interest of this syllabus and your curriculum, that is microbial spoilage of food. I have in the previous slides told you about the physical change, chemical change, now the microbial change. Microorganisms, they are omnipresent as they are present all over this slide. That is, I deliberately put all this over the slide because to make you understand, when a food is exposed to air, there is 100% chance that the air flora will be lodged on it. If your hands are not clean, we have seen the sources of contamination, put all that into act here, and you will see that the spoilage may be brought about by bacteria, yeast, molds, even viruses. All these are responsible for deteriorating the quality of the food. Up to some extent it can be poisoned also. But what happens is that as a food is a complete source of carbon, hydrogen, nitrogen, oxygen, phosphorus and sulfur, chnops, we can understand the urge of the microorganisms which are present in air. Air is not a natural medium for the growth of the organisms. When they lodge onto some food source, and if it is a very good medium, it has to be a very good medium for you to consume. That also makes it a very good medium for the microorganisms to grow and multiply, and as they multiply they go on deteriorating the quality of the food. A general pattern of the microbial spoilage, here you can see this three lines. There are three lines here. The straight line is total microflora, the big dotted lines are the changes, that is with respect to the shelf life, and small dotted lines are metabolites. We just concentrate on the total microflora, that is the log number of cells with respect to time. The shelf life, if you see, this goes on decreasing with time. Okay, there is a particular shelf life from 0 to this particular, you say 2 months, 3 months, that is the expiry date of the food. So above this particular time there is small dotted lines, they increase, that is the metabolites increase, because you can see the total microflora also increases. What this graph, general pattern of microbial spoilage, tells you is that the log number of cells after a particular period of time, that is which is called as a shelf life. How is this shelf life achieved? By adding preservatives. When preservatives are added in food, it does not allow the deterioration, but after certain time, that is 6 months, maybe for 1 year, after that there is a production of metabolites. First the production of metabolite gets enhanced because simultaneously there is a increase in the number of microorganisms. Concentration of metabolites are directly proportional to the log number of cells, and microbial spoilage takes place. What are the factors which affect the growth of the microorganisms? We have seen the factors, but again remember here, for causing spoilage in the food, intrinsic parameters, extrinsic parameters and implicit parameters are there. Intrinsic parameters: nutrients, water activity, pH, redox potential, inhibitors, and extrinsic parameter are temperature, humidity, atmosphere. Implicit parameters are interactions of one microorganisms with the other. Now, how does a microbial spoilage manifest itself? How does a microorganism allow itself to grow and spoil the food? That you can see by visible growth on the products, gas production is there, slimy layer is there, enzymes and off flavors and bad pungent odor. That is how you can just very easily conclude that food is spoiled. Now, having said that, there are various types of foods, there are various types of spoilages and the causing organisms, spoilage microorganisms, which is our main interest of discussion here. So if you see one of the most perishable foods, why I started with meat and poultry here. The first slide itself is meat and poultry. Why? Because this is one of the most easily perishable food as it is rich in nutrients. The meat, it can be fresh meat, cured meat or vacuum packed meat. With fresh meat, as you can see here in the table, fresh meat, putrefaction process takes place. This is Putrefaction is a process where proteins are degraded and very pungent smell of amino acid comes. So proteases are the enzymes responsible for putrefaction. As I always say, have a pen and paper and write it down. Those things which are not on the slide are also mentioned here. So putrefaction is degradation of proteins carried out by Clostridium, Pseudomonas, Proteus, alkali genes and Chromobacterium. Whereas souring of fresh meat, souring is khatta ho jana, sour, it becomes sweet and sour part. So Chromobacterium, Lactobacillus and Pseudomonas, they are responsible. Cured meat, what is cured meat? Which is subjected to some sort of processing, maybe drying, maybe heating or whatever it may be, that after some particular time gets spoiled, and that is moldy growth is due to fungi like Penicillium, Aspergillus, Rhizopus, etc. And souring of cured meat is due to Pseudomonas, Micrococcus, Bacillus, etc. Slime is polysaccharide material getting onto the meat. It becomes very slimy, means just like soap, and the responsible organism is Leuconostoc. In vacuum packed meat, souring and greening takes place due to Lactobacillus, Carnobacterium, Leuconostoc. You should be able to give the type of spoilage, the type of food and the causing organism in this example. Poultry, they have a bad odor and slime, which is mainly due to the organisms, microorganisms, Pseudomonas, alkali genes and Xanthomonas. In dairy, if you just go on to see the dairy, milk and cheese, these are the major products. Bitterness of milk, souring of milk and sweet curdling of milk, this takes place. Bitterness, kadusar hona, kadwa hona, this is due to Pseudomonas. Souring, khatta hona, that is due to Lactobacillus thermophilus. Sweet curdling, that is दूध का दही होना. Dahi, not just like the dahi which we desire, but a undesirable form, where the proteins they get separated. Dahi दूध फटना जो बोलते हैं वो, that is due to Bacillus cereus. Now cheese, there is green discoloration. Green is nothing but fungus. A fungus grows on this. Here in the picture you can see there is fungus growing on this cheese, that is due to Penicillium. And black coloration or green to black coloration may be due to Cladosporium. Total black may be due to Aspergillus. And sliminess due to high pH can be due to Pseudomonas species or Leuconostoc also. Gassy cheese, when you break up the cheese there will be gas bubbles inside the cheese and that will be very pungent, bad smelling, that is due to coliforms, lactic acid bacteria, LAB and Clostridia. Fish may get decolorized due to Pseudomonas. Putrefaction, the protein part may be degraded due to Chromobacterium, Halobacterium and Micrococcus. Eggs, they become rotten, green rot due to Pseudomonas. Pseudomonas is a pigmenting bacteria and its pigment gets dissolved in the medium where it grows. So if it is growing on egg, then it will make the egg green, that is called as a green rot. Colorless rot, that is Pseudomonas, alkali genes, Chromobacterium, they result into a rot. It is not colored but very bad smell is there. Black rot due to coliforms, and these coliforms they make the egg black due to hydrogen sulfide gas, H2S production. Fungal rot due to Proteus, Penicillium, Mucor, Aspergillus, etc. Then there is vegetables, fruits, canned food and beverages like wine. Fresh fruits and vegetables may have, you can pause this slide, I will not go on reading all the slides, pause here for a second and you can just go on reading as I have just explained all the types. The same applies to fresh fruits and vegetables, canned food and wine. There can be a off flavor in the wine, there can be a sour flavor in the canned foods, or there can be a gaseous swelling of the foods. All the reasons are there and the causing organisms is mentioned here. Now as I was saying about chemical changes, if the chemical changes are due to microorganisms, then it is caused by microorganisms which bring out the following changes. Degradation of carbohydrate takes place, degradation of nitrogenous compound takes place, degradation of lipids and pectin hydrolysis takes place. Degradation of carbohydrates, you can see alcoholic fermentation is there resulting into ethanol and carbon dioxide. Homofermentative lactic acid and heterofermentative lactic acid results into lactic acid, acetic acid, ethanol and carbon dioxide respectively. If it is propionic acid fermentation carried out by microorganisms by utilizing the substrates present in the foods, then propionic acid, acetic acid, carbon dioxide may be formed. Try to understand these acids and these products may be beneficial also, but if you have a controlled process or a controlled pathway carried out under your controlled set of conditions. But when it is taking place automatically, when it is uncontrolled, it crosses the boundaries of metabolism and end products are formed. Basically, what is the difference between a controlled process and an uncontrolled process? In controlled process, you know where to stop. If I want to produce citric acid, I know how to stop at citric acid, not allowed to go into isocitrate, malate or the oxaloacetate, so on and so forth. I know where to stop. But here naturally what happens, it goes into pyruvate, acetaldehyde, ethanol, carbon dioxide, and the whole food gets spoiled. So, like that, mixed acid fermentation, 2,3-butanediol fermentation, all these pathways by the microorganisms results into undesirable products. These products can be desirable if they are under controlled process. That you will study in the next semester in industrial microbiology. Try to understand, we are studying this in food spoilage. So automatic lodging of the bacteria on the food and metabolism which is uncontrolled results into these products which deteriorate the quality of the food. This is the pathway which I have been seeing, so I will not go into much detail of the pathway. This is the same slide as this slide which gives the product, but it is under a systematic flow sheet where it is showing all the products from glucose. How from glucose, oxaloacetate, propionate, butanediol, acetate, ethanol, hydrogen, carbon dioxide, formate, ethanol, butyrate, acetone, all these acids, they are a product of microbial metabolism or fermentation in the food utilizing the glucose or the carbon substrates present in the food. So here also you can see aerobic conversion of lactic acid to acetic acid by propanediol and 1,2-propanediol by lactobacillus buchaneri. This is one of the LAB group of bacteria, and it gives rise to acetic acid, acetaldehyde, lactaldehyde, ethanol. If these are needed, it is good, but if it is automatic, it is bad, so on and so forth. Here you can see the degradation of nitrogenous compounds. Proteolysis is one reaction where proteins are acted upon by proteinases. Proteinase is a group where we call it as proteases, and polypeptides and amino acids are resulting products. You can see proteins, these are acted upon by proteases which results into polypeptides. Polypeptides are acted upon by peptidases, and the peptide bonds are broken and it results into amino acids. This is called as proteolysis, and proteolysis is a reaction of a group of enzymes called as protease which is present in many bacteria. Amino acids, they can be, as you can see in this slide, amino acids cysteine, methionine, tryptophan, lysine, arginine, histidine, which are produced, these amino acids which are produced by proteolysis. Now these amino acids can be converted by the bacteria into cysteine, cysteine D-sulfahydrase to H2S. See the reaction. Amino acids are converted into volatile products by the bacterial cell. Cysteine is converted to cysteine is converted to hydrogen sulfide by the enzyme cysteine D-sulfahydrase. Methionine is converted to methyl mercaptan by the enzyme methionine lyase. Tryptophan is acted upon by tryptophanase to give rise to indole, whereas lysine is acted by decarboxylase. Lysine, arginine, histidine, they are acted by decarboxylase to give rise to cadaverine, putrescine, histamine. All these compounds, try to understand the dual mechanism of degradation of N compounds here. What is happening? Proteolysis is an enzyme reaction by protease where proteins are converted into amino acids, and amino acids by virtue of bacteria with the enzymes present, they are converted into volatile products. This is putrefaction. So degradation of N compounds, that is the proteinaceous compounds, nitrogenous compounds, takes place by proteolysis. This is called as putrefaction. Now reduction of thymine trimethylamine oxide, reduction of trimethylamine oxide. Where is this trimethylamine oxide present? Trimethylamine oxide is found in most of the seafoods. It is present in saltwater fishes. Trimethylamine is also normally found in foods such as soya beans, liver, kidney, wheat germ, brewer's yeast and egg yolk. So mostly in the animal foods which are coming from animal sources. And this trimethylamine oxidase, with the help of the enzyme trimethylamine oxidase reductase, is converted into trimethylamine, and this has a fishy odor. Even if it is a good food, it gives rise to a smell like a fish, सडलेल्या माशासारखं वास येतो. That is not desirable in the food. And which are the organisms which are responsible for this reaction? Pseudomonas, Schwannella, Bacillus, Clostridium. These are the bacteria which harbor the enzyme trimethylamine oxidase reductase, which converts the trimethylamine oxide to trimethylamine compound, which is fishy in odor. Now one of the most common reactions by the microorganisms responsible for the degradation of or spoilage of food is degradation of lipids. Lipids are acted upon by the enzyme lipase. Lipase converts lipids into glycerol and fatty acids. Fatty acids, they are further converted into aldehydes and ketones, and this gives rise to a very off flavor, of smell, and the food can be rejected by the consumer. This lipase is present in Pseudomonas, Micrococcus, Staphylococcus, Flavobacterium. Even in Bacillus it is present, which is present in soil. So these normal flora, these are the normal flora, these are not pathogenic organisms. They harbor these enzymes, and these enzymes are responsible for degradation of the food. Next, as you can see here, pectinases. Pectin degradation by pectinases. What happens is that example is apple, rotting of apple. Apple contains pectin. With the help of enzyme pectinases present in bacteria, fungi, etc., pectin is converted into polygalacturonic acid plus methanol. This polygalacturonic acid is converted to galacturonic acid, and this gives rise to a soft and watery type of either texture or dry and firm type of texture depending upon the microorganism. Penicillium expansum, here you can see here Penicillium expansum is responsible for soft and watery apple, whereas Monilinia fructigena is responsible for dry and firm type of food. So this is pectin degradation by microorganism. Slime, this is very important. It's a very simple pathway. Try to understand. Slime production is carried out by the bacteria when sugars are converted into slime, that is exopolysaccharides. Sugar is converted to glucose 6-phosphate. Glucose 6-phosphate is converted by certain enzymes which produce polysaccharides into uracil diphosphate glucose, UDP glucose, then uracil diphosphate galactose, UDP galactose, then thymidine diphosphate ramenose, DTDP ramenose. All these are exopolysaccharides. Exopolysaccharide is चिपचिपा जो होता है food, chip means slime, साबुन की तरह. That is the sliminess, and slime production is not desirable because most of the bacteria like leuconostoc and Pseudomonas, they are responsible for slime. Slime is a bacterial product, so definitely not desirable, and this results into ropiness also. Ropiness of the milk is very common. Again, I would like you to pause here, see this slide in a high quality mode, change the video quality here in YouTube, make it high quality 760 or 1080 HD mode. You can have this. Pause it here. You can see all the pathways of spoilage leading to the spoilage of wine. You can see here bitterness, then mousy of flavor, then you can see here the aroma change. All these are due to the change in the character of the solvent All these are brought about by various pathways. These are simple pathways: glycerol to acrotoin, glucose to glucan, fructose to mannitol. Then all these are responsible, because wine, it is aging of the wine, we have seen, takes long duration of time. It goes on for months together, and in those conditions, organisms like here as it is mentioned, acetobacter, lactobacillus, oniococcus, and pediococcus. I repeat: acetobacter, lactobacillus, oniococcus, pediococcus. These are the organisms which are responsible for bringing spoiled aroma and spoiled flavor compounds in wine. You can pause it and see the pathways. This off flavor or undesirable smell, all these are the types of spoilage responsible bacteria or the chemicals, and in which food it takes place. Fishy smell, trimethylamine, meat, egg and fish. In garlic, onion and cabbage, there is sulfide. Sulfur is there. Dimethyl sulfide, dimethyl trisulfide, and it is present as a component of wine, fish, milk, etc. And in this, the H2S production gives rise to a bad pungent smell. Fruity smell, it is the chemical compounds are esters, and that can be found in milk, fish and wine. The potato like flavor of meat and egg and fish, it is not potato, it is a potato like flavor in the meat or egg and fish, is due to 2-methoxy-3 isopropyl pyrazine, which is again not desirable because it is off flavor. Ethanolic flavor in fruit juices, mayonnaise. If you keep a fruit juice for long time, it gives ethanolic flavor because ethanol fermentation has taken place. Musty odor in bread and wine. Cheesy odor by meat, that is trichlorosenol, trichloroanisole, then diacetyl acetoin. Medicinal odor in juice and wine. There are certain juices which give a medicine like flavor. That is due to 2-methoxyphenol, which is a chemical produced due to some sort of metabolism which we saw in the last slide. Then soaring of any food, that is purely due to the acetic acid, lactic acid and citric acid. Acids are produced, organic acids are produced. Citric acid is sour, so citric acid is produced, lactic acid is produced, and it is a very common phenomenon in wine, beer and dairy industry. Texture problem, if you see the texture, physical change. Sliminess is due to polysaccharide. It is found in meat, juices, wine and confectionery. A bread or a piece of cake, if it becomes slimy, you should not consume it because there are certain bacteria which have produced polysaccharide. Softening of the foods is due to pectin degradation. We saw in apple example, phenolic decomposition. Fruits and vegetable, it is very common. Curdling is a texture problem which can be found in milk, and that is due to lactic acid production. Then cheese, holes in the cheese with gases, that is due to carbon dioxide. If you break up the cheese, there will be hole and there will be gases, CO2 gases, which is not desirable. Bloaters in fermented cucumber, holes in hard cheese, can swelling in canned foods. If there is a packed foods, it swells. You take a pack of Lay's or Kurkure, if it is extra swollen, some of the part air is there, that is a preservative gas present inside it, but it is extra swollen, that is due to gas swelling, and it may be any contaminant present in it. Finally, how to prevent it? It is very simple. If you are a microbiologist, you know how to take care of microorganisms. Keep the microorganisms out of the food. I am not saying keep the lactobacillus out of the curd. No. Keep the undesirable bacteria out of the food. Hinder their activity by lowering the temperature, keep it in the refrigerator, maybe drying, sun drying we use in ancient times, from ancient times, and chemicals, use of chemicals like pickles. What you do, you add extra amount of salt. Jams and jellies, you add extra amount of sugar, that itself becomes a preservative. Some antibiotics like maybe garlic is antibiotic compound, then your clove, lavang, that is also antibiotic compound. These can be added as spices and condiments. They are best antibiotics, natural antibiotics. They can be added in the food to keep the microorganisms at bay, to keep them away. You can even kill the microorganisms by subjecting it to heat. Finally, some important points of spoilage I would like to tell you. Usually we have these in detail in MSc level, but as you are students of graduation who aspire to become postgraduates or masters in this subject, I would like to throw some light on the present status of spoilage of food. Foods they spoil due to physical, chemical and microbial degradation, as we just saw, with the metabolites being the cause of off flavors, textural changes, or resulting into sensory rejection. These factors are interrelated to each other because at certain temperature, certain oxygen level, certain moisture concentration, the activity of the endogenous enzymes of the fruit itself, food itself, or the microorganisms present in the food which contain these enzymes, they are interrelated and they are put into action. Rodents and insect damage are also, see, we have not studied the last part which is insects like rats, rodents, lizards, mosquitoes, flies, etc. But remember the action of these rodents and insect damage. When a food is damaged by a rodent, it provides an entry point for the microbial growth, and microorganisms may gain entry, and it is just like an injury to the food and they can become infested. Which type of microorganism will develop, or what type of biochemical reaction occurs in which type of a food is dependent upon the type of the food and in which environment it is kept. That has to be remembered and that has to be sensibly cultivated. Although food spoilage is a major reason for economical loss, till now the food industry has not been able to 100% design a mechanism for preventing, because the underlying mechanisms are very poorly understood. You never know. Food may get spoiled by the food texture itself. Food may get spoiled by the microorganisms. Food may get spoiled by physico-chemical interactions. Food may get spoiled by the chemicals present inside the food itself. Like you see, a raw mango gets converted into ripe mango. It is automatic process. Chemical changes within. Ripe mango will get converted into a spoiled mango. No microbiology is involved. So you never know what is going on. So food spoilage is a very broad area. There is a need for identification and control of specific spoilage condition or organism. In microbiology it is called as SSO, specific spoilage organism, present in different food commodities, and it is very sad for the food industry that very less research is done in this, and specific spoilage organism for a specific food product is not established till date. Therefore the estimation of quality of food product still relies on the quantification of total numbers. What we do, we bring the food, see the total number of microorganisms and say if there are lot of microorganisms, this food is bad. That is true. Larger the amount of microorganism means that it has grown there. Why it has grown, and if it has grown, it is producing some chemicals, biochemicals, slime, etc. Getting the point? So in a food, what is the easiest way to determine the quality of a food? DMC, maybe SPC. Bring the food, see the number of microorganisms in food and say that more the number of microorganisms are there, more is the bad is the quality of this food. But this does not apply to those foods which are basically harboring microorganisms like lactic acid bacteria in curd. Got it? So finally I would like to say that the interaction between microbial spoilage and chemical spoilage must be understood very clearly, because a fruit or a food or a vegetable or a meat may undergo a chemical change without the need of a microorganism also. So you should not always jump to conclusion that a food spoilage is always due to microorganism. But as it is present, or we are studying here in microbiology, our perspective was and is microbial spoilage of food. So for the timing, this is all. My next lecture will be on microbial food poisoning, and pertaining to your syllabus we will be seeing 3 types of food spoilage. Thank you.